Hi everybody. Today we'll be looking into GATE Biotechnology 2017 paper, question number 17. The question is, during protein synthesis, tRNAs are not involved in option A, charging, option B, initiation, option C, elongation, and option D, termination. The central dogma of molecular biology states that DNA stores the genetic information that is passed on to the next generation. It is transcribed to the RNA, which is the dynamic functional molecule in the cell. This RNA is further translated into proteins. In this video, let us understand how translation works and how protein is synthesized to answer our question. So first of all, proteins are large complex molecules that play many critical roles in the body. They do most of the work in the cells and are required for the structure, function, and regulation of the body's tissues and organs. So, in other words, proteins are the structural and functional units of the cells and are responsible for any observable phenotypes, traits, and diseases. So, proteins are composed of one or more polypeptides. Polypeptides are linear chains of amino acids. Amino acids are molecules that are building blocks of the proteins. The sequence of amino acids in a polypeptide is known as its primary structure. So here we could see a simple polypeptide and we could see that two amino acids form this polypeptide. The two amino acids are linked together by a peptide bond. So the free amine end of this chain, polypeptide chain, is called the N terminus or the amino terminus, and the free carboxylic acid end is called the C terminus or the carboxyl terminus. So the formation of proteins from mRNA is called translation. It is also known as polypeptide synthesis or protein synthesis. It is a unidirectional process. That is, once proteins are formed, they cannot revert back to RNA as opposed to reverse transcription, in which the RNA can be converted back to DNA with the help of an enzyme called reverse transcriptase. So the raw materials for protein synthesis are amino acids, which is abbreviated as AAs throughout this video, and mRNA transcript, tRNAs or the transfer RNAs, and the amino acyl tRNA synthetases. So these are enzymes. So ribosomes catalyze the linking of the amino acid monomers directed by the mRNA sequence, and the amino acyl tRNA synthetases attach the amino acids to the appropriate tRNAs. So the amino acyl tRNAs, they act as adapters in the translation of the nucleic acid sequence of the mRNA into the amino acid sequence of the proteins. So let's look further into the details of how each of these components function in the process of translation. There are several molecules involved in translation, and let's look at the major ones. So amino acids, there are 20 different types of amino acids and amides which constitute the building blocks of proteins. And we have the mRNA molecule. So this carries the coded information for synthesis of one or more polypeptides. Its codons are recognized by the transfer RNAs. So tRNAs or transfer RNAs pick up specific amino acids and carry them over the mRNA strand. The amino acyl tRNA synthetase enzyme are specific for particular amino acids and their tRNAs. And finally, ribosomes, they have the amino acyl A and P, the peptidyl sites, are cavities on the ribosome where charged tRNA carrying the amino acids bind during polypeptide synthesis. The exit site is the site from which discharged tRNAs leave the ribosome. The mRNA binding site binds a sequence near the 5' end of the mRNA. The binding sites are all located at or near the interface between the large and small ribosomal subunits. So here's a pictorial representation of the tRNA and the ribosome. So the tRNA molecule has a distinctive folded structure with 3 hairpin loops that form the shape of a 3-leaf clover. So one of these hairpin loops contains a sequence called the anticodon, which can recognize and decode an mRNA codon. The other end is the amino acid receiving site where amino acids are attached and transferred to the growing polypeptide chain. And on the other hand, here's a ribosome. So this is the large and small subunit, and in the interface there's the mRNA binding site, and so we also have the AAP and the E sites on the ribosome. And we saw a term called codon. So the codon is on the mRNA and is recognized by the ribosome for translation. It is also called the genetic code that refers to the instructions contained in a gene that tells how to make a protein. So each group of 3 nucleotides on the mRNA is a codon. Since there are 4 bases A, U, G and C, there are 4 power 3, that is 64 possible codons which must code for 20 different amino acids. So more than one codon is used for more amino acids. So like more than one codon can be used to code for a single amino acid. So the genetic code is degenerate. So this means that it is not possible to take a protein sequence and deduce exactly the base sequence of the gene it came from, since more than one codon it codes for the same protein. So like if we have a amino acid leucine, it could have any of these codons, and hence we wouldn't know which exact sequence was there in the original DNA. So in most cases, the third base of the codon is called the wobble base, and it can be altered without changing the amino acid. And we could see that AUG is the start codon. So and it codes for methionine. So all proteins are initially translated with methionine in the first position, although it is often removed after translation. So the process of translation starts with methionine, and other amino acids are added later on. There are also internal methionines in most proteins coded by the same AUG codon. And there are 3 stop codons, which is UAA, UAG, and UGA. These are also called nonsense codons. So protein translation ends at the stop codon. So wherever there's any of these 3 codons, on the mRNA, the process of translation stops. So these codons, they do not code for any amino acid, and hence, the process of translation terminates. So these are the 64 different codons and the amino acids for which they code. And here's an example of an mRNA strand, and it could be read as UCG. So this could be a codon, or CGA could be a codon, and GAA could be a codon. So that depends on how the mRNA strand is read. So that is called a reading frame. And here we could see AUG, which is the start codon. So here, this is the place where the translation would start. And this region before the translation start site is called the 5' untranslated region. So this will not be translated to the protein. Only the protein will start from this region. For translation to happen, the tRNAs have to be charged. So charging happens in 2 steps. So in the first step, the amino acid and a molecule of ATP enter the active site of the enzyme aminoacyl tRNA synthetase. So the ATP loses the pyrophosphate, and this results in AMP, and this bonds covalently with the amino acid. So the pyrophosphate is then hydrolyzed into 2 phosphate groups, and then the tRNA covalently binds to the amino acid to displace the AMP, and the aminoacyl tRNA is then released from the enzyme. So the aminoacyl tRNA is the tRNA attached to the amino acid. So now this tRNA is charged with the amino acid. Now getting into the process of translation. A prokaryotic mRNA molecule encoding a single polypeptide has a 5' non-coding leader sequence which contains a ribosome binding site. The rest of the mRNA contains a coding sequence that starts with an AUG start codon and ends with a stop codon and a 3' non-coding trailer sequence. These are also called the untranslated regions. So a eukaryotic mRNA, on the other hand, in addition to the above sequences, has a 5' cap and a 3' poly A tail. And one important difference to note between that of the prokaryotic mRNA is that the eukaryotic mRNAs lack a ribosome binding site called a Shine-Dalgarno sequence, which we'll come back to in the next few slides. So N-formyl methionine, abbreviated as FMET, is the modified amino acid with which every polypeptide is initiated in prokaryotes. But in eukaryotes, methionine is the first amino acid. So there are, just like in transcription of mRNAs, there are three steps in protein translation. So they are initiation, elongation, and termination. So in initiation, the components of translation interact with the mRNA transcript. A transfer RNA carrying the first amino acid, which is methionine or FMET, binds to the start codon in the mRNA. And during elongation, the amino acids are brought to the mRNA as amino acyl tRNAs are added one at a time to the growing polypeptide chain. And finally, in termination, a stop codon in mRNA is recognized by a protein release factor, and the translational apparatus comes apart, that is, dissociates, to release a complete polypeptide or a protein molecule. Now let's look further into each of these steps. So first is the initiation of translation. So in prokaryotes, the formation of the 70S translation initiation complex occurs in three steps. So three initiation factors abbreviated as IF, so IF1, IF2 and IF3 along with GTP bind to the small ribosomal subunit. So the initiator amino acyl tRNA and mRNA are attached. The mRNA binding site is composed of a portion of the 16S rRNA of the small subunit. The 3' end of the 16S rRNA has a pyrimidine-rich sequence that binds with the Shine-Dalgarno sequence of the mRNA. So this Shine-Dalgarno sequence of the is typically found around 4 to 7 bases upstream of the translation start codon on the mRNA and has a sequence AGG AGG. So this sequence is complementary to a part of the 3' end of the 16S rRNA. So this portion is complementary to the Shine-Dalgarno sequence. So the large ribosomal subunit then joins the complex. So the resulting 70S initiation complex of FMET, tRNA, FMET resulting in the ribosome's P site. The initiation in eukaryotes, on the other hand, is similar to that in prokaryotes except for a few differences. So the eukaryotic mRNAs do not have a Shine-Dalgarno sequence which is recognized by the ribosome. So the ribosomes in the eukaryotes, they bind to the 5' cap, then move down the mRNA until they reach the first AUG, that is the start codon which codes for methionine. So translation starts from this point, and eukaryotic mRNAs code for only a single gene. So also note that the translation does not start at the first base of the mRNA. So this, like we saw, is called the 5' untranslated region. So the initiation process, like we just saw, involves the joining of the mRNA, the initiator methionine loaded tRNA, and the small ribosomal subunit. So here we have several initiation factors, 1 to 6, which are additional proteins involved. So in the prokaryotes, we just had 3 initiating factors, while in eukaryotes, we have 6. So the large ribosome subunit then joins the complex. Now moving on to the next step, which is elongation. Elongation begins with the binding of the second amino acyl tRNA at the ribosomal A site. The tRNA is escorted to the A site by the elongation factor EFTU, which also carries 2 bound GTP molecules. As the tRNA binds, the GTPs are hydrolyzed and EFTU is released and recycled by the elongation factor TS. A peptide bond is formed between the carboxyl group of the terminal amino acid FMET in the first cycle at the P site and the amino acid group of the newly arrived amino acid at the A site. This reaction is catalyzed by the peptidyl transferase activity of the 23S rRNA in the large ribosomal subunit. So after EFG GTP binds to the ribosome and GTP is hydrolyzed, the tRNA carrying the elongated polypeptide translocates from the A site to P site. The discharged tRNA moves from the P site to the exit site, which is the E site, and it leaves the ribosome. As the peptidized tRNA translocates, it takes the mRNA with it. So the mRNA also moves. The next codon is moved to the A site, which is now open for the next amino acid tRNA to bind. So these steps are repeated for each additional amino acid. So the elongation process in eukaryotes is similar to that of prokaryotes, except that the different elongation factors EF1 and EF2 are used. So the final step in translation is termination. So termination depends on release factors called RFs. So these release factors recognize the stop codons on the mRNA. So when a stop codon arrives at the A site, it is recognized and bound by a protein release factor RF. So this causes the polypeptide to be transferred to a molecule of water to cause its release from the tRNA and the dissociation of the other components of the elongation complex. So the protein is finally released from the whole transcription complex, and this is the end of the translation. So the eukaryotic termination is also similar, except that ERF1 release factor is used. Also, the mRNA continues beyond the stop codon. That is, there is a stretch of mRNA beyond the stop codon, and this remaining portion is not translated, and it's called the 3' untranslated region or 3' UTR. Let us now look at a short video to understand better about translation.
The second step of translation is called elongation because our peptide is going to start getting longer. After the initiator transfer RNA attaches to the P site, the second transfer RNA, which is carrying another amino acid, settles into the A site on the ribosome. If the second codon is GCU, for example, the second transfer RNA will have an anticodon of CGA. This particular transfer RNA would be carrying the amino acid alanine. An enzyme in the large subunit of the ribosome transfers the methionine from the initiator transfer RNA to the alanine bound to the second transfer RNA. A peptide bond forms between these two amino acids, forming a dipeptide. Once this happens, the ribosome slides on to the next codon on the messenger RNA. This action shifts the initiator transfer RNA to the E site, and the second transfer RNA to the P site. Having given its amino acid to the dipeptide, the initiator transfer RNA will exit the ribosome and seek out another methionine in the cytosol. The next transfer RNA will settle into the A site. Once again, the enzyme transfers the amino acid at the P site to the transfer RNA at the A site. However, this time, both amino acids of the dipeptide transfer over, making the peptide an even longer chain of amino acids. This process continues until the ribosome reaches the stop codon, at which point a protein called release factor binds to the A site rather than transfer RNA. The new protein releases from the ribosome and the ribosome disassembles. This last step is called termination, and our new protein is now ready for packaging and export.
After translation, there are several things that happen to the new protein molecule, and this is called post-translational modification. So new polypeptides usually fold themselves spontaneously into their active conformation. Many proteins have sugars, phosphate groups, fatty acids, and other molecules which are covalently attached to certain amino acids. So most of this is done in the endoplasmic reticulum of the cell. So many proteins are targeted to specific organelles within the cell. Targeting is accomplished through signal sequences on the polypeptide. In the case of proteins that go into the endoplasmic reticulum, the signal sequence is a group of amino acids at the end terminal of the polypeptide, which are removed from the final protein after translation. So this is an overview of the whole process of translation. Now, coming back to a question, which is, during protein synthesis, tRNAs are not involved in which of the following steps? So option A, charging. So we saw that the tRNA has to be charged with the amino acid for translation to start. So tRNA is involved in charging, and so this is not the answer. And option B, initiation. So during initiation, tRNA has to carry the first amino acid, which is methionine, to the mRNA. And so B is not the answer as well. And we have option C, elongation. And we just know that tRNA plays a role in elongation, which carrying different amino acids to the translation complex. So C is not the answer. This leaves us with option D, termination. So termination does not actively involve tRNA. So the tRNA is dissociated from the whole complex. So the process of termination only requires the release factors. So the answer would be termination. So during protein synthesis, tRNAs are not involved in termination. Thank you.